This is USMLE QA, USMLE material presented in a question and answer format structured similar to the first aid for the USMLE Step 1 book.
Question: what are the steps of protein synthesis? Answer: protein synthesis consists of several stages, including initiation, elongation, and termination. Question: can you explain the initiation stage of protein synthesis? Answer: the initiation stage of protein synthesis begins when a specific sequence of nucleotides in the DNA, called a promoter, is recognized by enzymes called RNA polymerase. This initiates the process of transcription, during which a complementary RNA strand is synthesized. Question: how does the elongation stage of protein synthesis work? Answer: the elongation stage of protein synthesis occurs when the RNA polymerase moves along the DNA template, adding nucleotides to the RNA strand. This process is guided by the base pairing rules of A-U and C-G. Question: can you explain the termination stage of protein synthesis? Answer: the termination stage of protein synthesis occurs when the RNA polymerase reaches a specific sequence in the DNA called a terminator. This signals the polymerase to stop transcribing the RNA and release it from the DNA template. The newly synthesized RNA strand can then be processed and translated into a protein. Question: how does the process of translation begin after protein synthesis? Answer: after the RNA strand is synthesized and released from the DNA, it is transported to the ribosomes where the process of translation begins. The RNA strand is read in triplets called codons, which correspond to specific amino acids. Question: can you explain the role of transfer RNAs in protein synthesis? Answer: transfer RNAs play a crucial role in protein synthesis by carrying specific amino acids to the ribosome during translation. Each tRNA molecule has a specific anticodon that base pairs with a codon on the mRNA strand, bringing the correct amino acid to the ribosome. Question: what is the role of the ribosome in protein synthesis? Answer: the ribosome serves as the site of protein synthesis during translation. It reads the codons on the mRNA strand and uses the information to bring the correct amino acids together and link them through peptide bonds. This process results in the formation of a polypeptide chain, which will eventually fold into a functional protein. Question: can you explain the process of protein folding? Answer: protein folding is the process by which a polypeptide chain, formed during translation, takes on a three-dimensional shape. This shape is determined by the sequence of amino acids and the interactions between them. Protein folding occurs spontaneously, but is assisted by chaperone proteins that help guide the polypeptide into its correct shape. Proper folding is essential for the protein to function correctly. Question: how does protein synthesis end? Answer: protein synthesis ends with the release of the newly formed protein from the ribosome. The protein may then undergo further modifications such as the addition of sugar or lipid molecules, or be transported to a specific location in the cell where it will carry out its function. This completes the process of protein synthesis from initiation to termination. Question: what are the main factors that affect the rate of protein synthesis? Answer: the rate of protein synthesis can be affected by several factors, including the availability of the necessary components, the activity of enzymes involved in the process, and the presence of regulatory factors that control the rate of protein synthesis. Question: what is the role of mRNA in protein synthesis? Answer: mRNA plays a crucial role in protein synthesis as it carries the genetic information from the DNA to the ribosomes where it is translated into a protein. The mRNA strand is synthesized during the process of transcription and serves as a template for the synthesis of the protein. Question: what is the difference between transcription and translation? Answer: transcription and translation are two distinct stages of protein synthesis. Transcription is the process of copying the genetic information from DNA to RNA, while translation is the process of converting the information on the RNA into a protein. Transcription occurs in the nucleus, while translation occurs in the cytoplasm. Question: what are the different types of RNA involved in protein synthesis? Answer: three different types of RNA are involved in protein synthesis: messenger RNA, transfer RNA, and ribosomal RNA. mRNA carries the genetic information from the DNA to the ribosomes. tRNA brings the correct amino acids to the ribosome during translation, and rRNA is a component of the ribosome that helps to read the mRNA and link the amino acids together. Question: how does protein synthesis differ between prokaryotic and eukaryotic cells? Answer: protein synthesis in prokaryotic cells differs from that in eukaryotic cells in a few key ways. In prokaryotic cells, transcription and translation occur in the same location, the cytoplasm, while in eukaryotic cells, transcription occurs in the nucleus and translation occurs in the cytoplasm. Additionally, prokaryotic cells have a single circular chromosome, while eukaryotic cells have multiple linear chromosomes. This can affect the way in which the genetic information is accessed and processed during protein synthesis.
Thank you for watching our Q&A format video. We hope this helped you learn, review, and retain knowledge needed for the USMLEs. For more information on this topic, check out the link in the description. Now, if you want to take your USMLE prep to the next level, be sure to check out all our courses, which are structured to follow along with the First Aid for the USMLE Step 1 book. You can find us at usmleqa.com. And if you're watching this on YouTube, be sure to like and subscribe.